Okay, let's unpack this. Imagine you're a plant. You're stuck, right? Rooted in the soil. How do you possibly get the specific nutrients, the ions you need from right there? And then how do you move them around? It seems like a huge challenge.
Oh, it absolutely is. And it's definitely not just simple absorption, getting specific molecules, specific ions across that cell boundary, the membrane, and then getting them where they need to go inside the plant. That takes some seriously sophisticated machinery.
Right. And that machinery, those processes, that's what we're diving into today. Solute transport in plant cells, how stuff moves in, moves out, and moves around within the plant.
Exactly. And we're using a key chapter from a pretty detailed plant physiology textbook as our guide here. Think of us as helping you navigate through the core ideas of the physics, the molecules involved.
And why it all matters, fundamentally, for how plants actually live. Because, yeah, this isn't just textbook stuff. It's key to understanding plant growth, how they get the minerals that, end up in our food, and also how they cope with stress, like salty soil. If we want to feed everyone sustainably, we really need to get this.
So our mission today is to break down this dense material, make it clear, make it engaging for you, show the how and the why of plant transport.
So where do we start? The boundary.
Yeah, let's start right at the cell membrane. It's this super thin layer, mostly lipids, separating the inside world of the cytosol from the outside. It's a barrier, sure, but it has to let things through. Selectively.
A selective gateway. And the movement across it, or movement elsewhere that's controlled by it, that's transport.
Right. And it happens on different scales. Locally, across one membrane, maybe between cells right next to each other. But also long distance, think sugars made in a leaf going all the way down to the roots through the phloem. That long-haul trip still depends on transport events at the cellular level.
Okay, so what makes things move? What are the driving forces? We've talked about concentration gradients before, right? And pressure.
Yes. And for ions, things with a charge, electrical gradients are just as important, sometimes more so. Gravity, not really a big factor at this tiny scale.
So concentration and electrical forces are the main players. How do we combine those ideas?
Okay, so for molecules without a charge, like sugar, sucrose, the driving force is summed up by its chemical potential. Basically, it's the free energy that a molecule has available to move.
Like potential energy for movement.
Kind of, yeah. And things naturally move from high chemical potential to low chemical potential. For these uncharged solutes, it mostly comes down to the concentration difference, really, the ratio of concentrations inside versus outside.
So if there's way more sucrose outside than inside, its chemical potential is higher outside, and it will want to move in, assuming it can get through the membrane, of course.
Exactly right. And that movement downhill, following the chemical potential gradient, that's passive transport. The cell doesn't have to spend metabolic energy. It's like rolling downhill.
Okay. But plants often need to move stuff uphill, right? Against the gradient.
They certainly do. Moving something from low concentration to high concentration against its chemical potential gradient, that's active transport. It won't happen on its own. It needs energy, usually from the cell breaking down ATP.
Got it. Passive is downhill, no extra energy needed. Active is uphill, needs fuel like ATP. Simple enough. But what about ions, the charged stuff?
Right. For ions, it gets a bit more complex. You have to consider both the concentration difference and the electrical potential difference, the voltage across that membrane.
Ah, okay.
This combined driving force is called the electrochemical potential. So for a positive ion, like potassium, K+8, having a high concentration outside pushes it in. But also, if the inside of the cell is electrically negative compared to the outside, that negative charge pulls the positive K+ ion in too. Both factors contribute.
So the voltage can help pull ions in, even if the concentration inside is already pretty high.
It absolutely can. So ions move down their electrochemical gradient. Passive transport for ions means following that combined gradient downhill. Active transport means pushing them against it uphill, which, again, needs energy.
Okay, that makes sense. How easily do things actually cross the membrane, though? What determines that?
That's membrane permeability, how readily the membrane lets something pass through. The basic lipid bilayer lets some small uncharged things slip through, okay?
Like water, maybe?
Water, some gases, yeah. But for ions and bigger polar molecules like sugars or amino acids, the permeability depends almost entirely on specific transport proteins embedded within that membrane.
Ah, the proteins are the gatekeepers.
They are. And permeability affects the rate how fast things move. It doesn't change the final direction or the equilibrium point.
Yeah.
Equilibrium happens when the electrochemical potential difference hits zero across the membrane.
Okay. Now, you mentioned voltage. How does the movement of these ions actually create that voltage across the membrane?
Good question. Biological membranes can build up a voltage, a membrane potential, because they aren't equally permeable to all ions. And ions move at different speeds. Let's say a membrane is much more permeable to potassium, K plus Li, than to chloride, Cl(D). If there's more K plus inside, it will tend to diffuse out faster than Cl diffuses in.
So positive charge leaves faster.
Right. Even a tiny net movement of positive charge out leaves the inside slightly negative relative to the outside. That separation of charge, however small, creates a voltage, a diffusion potential. In many plant cells, K plus is quite mobile, so its movement is a big factor in setting the membrane potential, often making the inside negative.
But does this mean the solutions themselves become charged? Like the whole cytoplasm gets negative?
No, that's a key point. The bulk solutions on either side stay electrically neutral. The charge separation that creates the voltage is incredibly small, localized right at the very surface of the membrane. You might only need, say, one extra negative ion inside for every 100,000 ions to create a negative 100 millivolt potential. It's chemically tiny, but electrically significant.
Wow, okay. So what if, hypothetically, a membrane was only permeable to one single type of ion?
In that specific case, that ion would move across the membrane until the electrical potential difference perfectly balanced its concentration difference. At that point, the net driving force, its electrochemical potential difference, would be zero.
It reaches equilibrium.
Exactly. And the specific voltage at which that balance occurs is called the Nernst potential for that ion. The Nernst equation gives you the math for it.
Is there a simpler way to think about it?
Yeah, there's a handy rule of thumb. For an ion with a single charge, like K plus or Cl at room temperature, a 10-fold difference in its concentration across the membrane corresponds to a Nernst potential of about 59 millivolts.
Okay, useful shortcut. And can we actually measure the real voltage in a living cell?
We can, yeah. Scientists use incredibly fine microelectrodes, carefully inserted into a single plant cell, to measure the voltage difference between the inside and the outside. Plant cells typically show a resting potential somewhere between negative 60 and negative 240 millivolts, usually quite negative inside.
So if we measure that voltage, and we know the ion concentrations inside and out, we can use the Nernst equation
to predict what the internal concentration should be if that ion were just passively distributed, just balancing its concentration and the electrical potential.
And then compare that prediction to what we actually measure inside the cell.
Precisely. If the measured internal concentration matches the Nernst prediction, the ion might be at equilibrium, but often it's not.
Because of active transport messing with the equilibrium.
Exactly. Living cells are constantly pumping ions around, using energy to move them against their electrochemical gradients. This creates a steady state where concentrations are stable over time because influx equals efflux, but it's not necessarily equilibrium. Active pumps are balancing passive leaks.
Can you give an example?
Sure, that P-root cell data in the textbook is great for this. They measured the membrane potential at negative 110 milliV. Then they looked at various ions for anions like nitrate, phosphate, chloride. The actual concentration inside the root cells was much higher than the Nernst equation predicted for passive distribution at negative 110 milliV.
Meaning?
Meaning the cell must be actively pumping those anions in, working against the electrochemical gradient that would otherwise push them out.
And for positive ions, cations.
For cations like sodium, Na+, and calcium, say A2+, the measured internal concentrations were much lower than the Nernst prediction. This implies they passively leak in, down their steep electrochemical gradient, but the cell actively pumps them out or sequesters them in compartments like the vacuole to keep the cytosolic levels low.
Okay, so the Nernst equation is like a diagnostic tool for active transport. But you said plant potentials can be really negative, like minus 200 milliV, often more negative than simple ion diffusion, even considering multiple ions would suggest. What causes that extra negativity?
That's where electrogenic pumps come in. These are transport proteins that don't just move ions, they move a net electrical charge across the membrane as part of their cycle. This charge movement directly generates an electrical potential.
And in plants, the main one is?
In plants, also fungi and bacteria, the star player on the plasma membrane is the plasma membrane H plus ATPase, the proton pump.
Right, the proton pump.
It uses energy from ATP to actively pump protons, hydrogen ions, H plus out of the cytosol into the cell wall space. Pumping positive charge out makes the inside significantly more negative. This is what drives the membrane potential to those very negative values, well beyond what passive ion diffusion alone could create.
Is there direct proof for that?
Oh yeah, classic experiments use metabolic inhibitors like cyanide, which block ATP production. When you add cyanide, the membrane potential rapidly becomes much less negative. It collapses toward the potential you'd expect just from passive diffusion.
Because the pump runs out of fuel.
Exactly. And at the same time, you see protons, H+, building up inside the cell, making the cytosol more acidic, while the outside becomes more alkaline. Clear evidence that the pump was actively extruding protons using ATP.
So this proton pump is like the main engine generating the negative voltage.
It's a huge contributor, yes. It sets up this strong electrical gradient.
And how does that strong negative potential affect other ions then?
Well, think about positive ions. Cations like potassium, K+. That strongly negative inside acts like a magnet, providing a powerful electrical driving force, pulling K+ into the cell passively through any open potassium channels, even if K+ concentration inside is already substantial.
So it helps the plant take up essentialitations passively?
It facilitates passification uptake, exactly. And these proton gradients are vital elsewhere too. Think ATP synthesis in mitochondria and chloroplasts, which relies on H+ gradients.
Okay, we've got the forces, the gradients, the main pump setting the electrical scene. Let's talk about the specific proteins that actually move things across, the molecular machines.
Right. If you compare a real biological membrane to just a plain artificial lipid bilayer, the difference is stark. The artificial one is pretty impermeable to ions and most polar stuff. Biological membranes let these things through much more readily.
Because of the transport proteins.
Because they're packed with specific transport proteins. The textbook highlights three main classes: channels, carriers, and pumps.
The big three.
Yep. And they are typically very specific for what they transport. Plants have a huge number of genes coding for these, maybe around 1,800 transporter genes in Arabidopsis, which isn't even a complex plant.
Wow, thousands of different doorways. Let's start with channels. What are they like?
Channels are basically selective pores or tunnels through the membrane. When they're open, they allow specific ions, or sometimes small neutral molecules, to diffuse through very rapidly, always moving down their electrochemical gradient.
So always passive transport.
Always passive for channels. Their specificity comes from the size of the pore and the chemical properties, like electrical charges, lining the inside of that pore.
And you said they're fast.
Incredibly fast when open. Millions, even hundreds of millions of ions can pass through a single open channel per second, like opening a floodgate.
But they're not always open, are they?
No. Critically, they have gates. These gates open and close in response to specific signals, which allows the cell to regulate permeability very quickly.
What kinds of signals?
All sorts. Changes in the membrane voltage itself, those are voltage-gated channels. Binding of a specific molecule, like a neurotransmitter or hormone ligand-gated channels. Some respond to light, mechanical stretch, or chemical modification like phosphorylation.
So they're highly regulated switches.
Absolutely. And there's huge diversity, many different types of potassium channels, for instance. Some open when the membrane gets very negative inside, letting K+ flow in. They're called inwardly rectifying. Others open when it's less negative or even positive inside, letting K+ flow out, outwardly rectifying. These are vital for things like controlling stomatal opening and closing in leaves. You also have channels for anions, calcium, and others.
Okay, channels. Fast, passive, gated pores. What about carriers? How are they different?
Carriers work more like a revolving door or an airlock. They don't form a continuous open pore. Instead, they bind to the specific solute molecule on one side of the membrane.
Like an enzyme binding its substrate?
Very similar analogy, yes. They have a specific binding site. After binding, the carrier protein undergoes a conformational change. It changes its shape, which moves the solute across the membrane and releases it on the other side.
So it involves a shape change for every molecule moved.
Right, which makes them much slower than channels, maybe hundreds or thousands of solutes per second, not millions. But that binding step also makes them highly specific.
And can carriers do active transport?
They can mediate passive transport, which is called facilitated diffusion, still moving downhill, just using the carrier to get across. But importantly, carriers are also responsible for secondary active transport.
Okay, we'll come back to that. That leaves the third type, pumps.
Pumps are the primary engines of active transport. They directly use a source of energy, typically ATP hydrolysis in plants, to force solutes against their electrochemical gradient. Uphill movement.
Like the proton pump we already discussed.
Exactly. The plasma membrane H+ ATPase is a classic pump. Other examples include pumps that specifically move calcium ions. CO2 plus He bases, and a very large family called ABC transporters, which use ATP to move a huge variety of molecules, often larger organic ones, across membranes. They are directly fueled.
Okay, you mentioned carriers and secondary active transport, and earlier you talked about the proton pump creating this big H plus gradient, the proton motive force. Do those connect?
They absolutely connect. Secondary active transport is all about harnessing the energy stored in an existing gradient, usually the proton gradient, PMF, set up by the H plus ATPase in plants.
So a cell spends ATP to pump protons out, creating the PMF.
And then carrier proteins use the energy release when those protons flow back in down their gradient to drive the uphill movement of another solute.
Clever, using one gradient to build another.
It's very efficient. There are two main flavors of this: symport and antiport. Symport means transport together. The carrier moves both the driving ion, like H+, and the transported solute in the same direction across the membrane. For instance, a proton flows downhill into the cytosol, and the carrier couples that movement to drag, say, a nitrate ion or a sugar molecule into the cytosol along with it, even if that nitrate or sugar is moving against its own gradient.
So H plus flowing empowers uptake of something else. What's antiport then?
Antiport means transport opposite. Here, the carrier moves the driving ion, like H+, in one direction, and the transported solute in the opposite direction. So a proton might flow downhill into the cytosol, and the energy released is used to push a sodium ion, No+, or a calcium ion, Cl2+, out of the cytosol against its gradient.
Ah, so symport is often for uptake, antiport often for export or sequestration.
That's a common pattern, yes. H+ symporters bring in nutrients like nitrate, phosphate, amino acids, sugars. H+ antiporters push out toxic ions like sodium or move things into the vacuole. It's this constant circulation. H+ pumps push protons out, and secondary transporters let them back in, coupling that flow to move other essential molecules.
This sounds incredibly complex. With potentially thousands of transporters, how do researchers even figure out which protein does what?
Yeah, it's a big challenge. They use various techniques. Sometimes they find mutant plants that can't transport something properly and then identify the affected gene. Or they take the plant gene and express it in something simpler, like yeast cells or phagocytes, and see if that cell gains the ability to transport the substance. Comparing gene sequences helps group transporters into families with likely related functions. It's a huge area of research.
And this leads to finding transporters for basically everything a plant needs.
Pretty much. Take nitrogen. Plants need a lot of it. They take it up mainly as nitrate, NO3, or ammonium, NH4+. Nitrate uptake involves H+ symporters, and it's complex. There are systems that work best at low nitrate concentrations, high affinity, and others for high concentrations, low affinity. Some transporters, like one called CHL1, are amazing. They act as both a transporter and a sensor for nitrate levels.
Wow. What about ammonium?
Ammonium uptake seems to be mainly through facilitator-type channels, moving downhill passively. Then you have transporters for amino acids, small peptides, often H+ importers, again crucial for moving nitrogen around the plant, especially from storage tissues or during senescence. Even carnivorous pitcher plants use peptide transporters to absorb nutrients from digested insects.
And those ABC transporters you mentioned.
Huge family. They use ATP directly to transport an incredible variety of things, not just peptides, but also hormones like ABA, pigments like anthocyanins that give flowers color, secondary metabolites for defense, often moving them into the vacuole for storage or detoxification.
Okay, what about positive ions, K-phations? You mentioned potassium channels.
Right. Potassium K+ is vital. Plants have various channels for K+ uptake and release, like the Shaker family channels involved in root uptake, xylem loading, guard cell function. There are also carrier systems like the HAK family for high-affinity K+ uptake, often working as H+-K+ importers, and others involved in moving sodium, calcium, magnesium.
And this ties into things like salt tolerance, you said.
Yes, absolutely. Dealing with excess sodium, Na+, is critical for plants in salty soils. They rely heavily on specific transporters. One key player is SOS1, a Na plus H+ antiporter on the plasma membrane of root cells that pumps sodium out of the cell.
Using the proton gradient.
Yes, H+ flows in, Na+ flows out. Then there's another important one, NHX1, which is an A plus H plus antiporter located on the vacuole membrane, the tonoplast. It pumps sodium into the vacuole, safely sequestering it away from the sensitive enzymes in the cytosol.
So pump it out of the cell or lock it up in the vacuole.
Exactly. And increasing the activity of these transporters, for example by genetically engineering plants to make more NHX1, has been shown to significantly improve salt tolerance in crops like tomatoes, rice, even wheat. Another transporter, HKT1, Helps by removing sodium from the water stream, xylem, heading to the leaves, protecting them. These are really promising avenues for developing salt-resilient crops.
That's a fantastic real-world application. What about calcium? You said it's kept really low in the cytosol.
Extremely low. Calcium acts as a crucial secondary messenger, a signal. So when a signal needs to be sent, calcium channels open briefly, letting Ca2 plus flood in passively down its very steep electrochemical gradient.
Creates a quick spike.
Right, but then that signal needs to be turned off quickly. So cells have powerful active pumps, C2 plus ATPases and Ke2 plus HLH plus antiporters on the plasma membrane and internal membranes, like the ER and the tonoplast, that rapidly pump the calcium back out of the cytosol or into storage compartments like the vacuole. It's tightly controlled. Proteins like calmodulin bind to the incoming calcium and then help regulate the channels and pumps, providing feedback.
Okay. Anions besides nitrate, like phosphate.
Phosphate, H2PO4, is often scarce in soils, so plants have high-affinity H+ importers in their roots to scavenge it effectively. These are often induced when phosphate is low. Chloride, sulfate, malate, they all have specific transporters, often H+ importers for uptake against their gradients, and channels often involved in efflux.
Metals, micronutrients.
Yep, essential metals like iron, zinc, manganese, copper are taken up by specific transporters like the ZIP family. Some of these transporters can also unfortunately take up toxic heavy metals like cadmium, which is relevant for both food safety and potentially using plants for phytoremediation, cleaning up contaminated soils. Plants also have mechanisms to transport these metals internally, often bound to chelating molecules, and detoxify excess amounts, often by sequestering them in the vacuole.
What about things like boron or silicon?
Boron, as boric acid, and silicon, as silicic acid, are technically metalloids. They actually enter plant cells, at least partly, through certain types of aquaporins, the water channels.
Water channels move boron and silica.
Some specific aquaporins, yes. They seem permeable to these small, uncharged molecules. Efflux, getting them out again or loading them into the xylem, involves different specific transporters. Interestingly, arsenite, a toxic form of arsenic, can sneak into rice roots through silicon channel cycloporins, which is a major health concern in regions with arsenic-contaminated water.
So aquaporins aren't just for water.
Primarily, yes, they facilitate massive water flow. But some isoforms clearly transport other small neutral solutes like boric acid, silicic acid, maybe even ammonia, CO2, or hydrogen peroxide. Aquaporin activity itself is highly regulated. Phosphorylation, pH changes, calcium levels can all affect their opening and closing, allowing plants to rapidly adjust water permeability in response to drought or other signals.
You mentioned the vacuole membrane, the tonoplast, having transporters like the NHX antiporter. Does it also have a primary proton pump like the plasma membrane?
It does. Actually, it has two main types of proton pumps that push H+ into the vacuole, making the vacuole acidic, low pH, and electrically positive relative to the cytosol.
Pumping H+ in this time.
Yes. One is the V-ATPase, or vacuolar H+ ATPase, which uses ATP. The other is the H+ P-pase, or vacuolar proton pyrophosphatase, which uses pyrophosphate, PPI, another energy-rich molecule, as its fuel.
So the vacuole builds its own proton gradient.
Exactly. And this vacuolar proton gradient, this PMF across the tonoplast, is then used to power secondary active transport into the vacuole. Lots of H+ antiporters on the tonoplast use the outward flow of protons back into the cytosol to drive the import of ions like Na plus T2 plus, sugars, amino acids, and waste products into the vacuole for storage or detoxification.
Okay, so proton pumps on both the plasma membrane, pumping out, and the tonoplast, pumping in, create gradients that drive a whole suite of secondary transport processes.
Precisely. It sets up different compartments with distinct environments and transport capabilities.
Let's zoom out now. How does all this cellular transport work together in a whole tissue, like getting ions from the soil into the root and then up the plant?
Okay, think about a root in the soil. There are two main pathways for water and solutes to move radially inwards towards the center.
Two paths.
Yes. One is the apoplast pathway. This is the interconnected network of cell walls and the spaces between cells. It's like a wet sponge water.
Yeah.
And dissolved solutes can just diffuse through this non-living space passively.
So just soaking through the walls.
Pretty much, yeah, following the flow of water. The other pathway is the simplest. This is the interconnected cytoplasm of all the living root cells, connected by little bridges called plasmodesmata that pass through the cell walls.
So once an ion crosses a plasma membrane to get into one cell,
it can then move from cell to cell through these cytoplasmic connections, through the simplest, without having to cross it on membrane.
Okay, apoplast through the wall, simplest through the cytoplasm. Where's the control point? Does the plant just let anything soak in through the apoplast?
No. There's a crucial checkpoint. It's a layer of cells deep inside the root called the endodermis. And the endodermal cells have a special feature in their walls called the Casparian strip.
The Casparian strip, what does it do?
It's a band, like a gasket, made of waterproof material, suberin and lignin, embedded within the cell walls of the endodermis. It completely blocks the apoplast pathway at that point. Water and solutes moving through the cell walls hit this waterproof barrier and can't go any further through the wall.
So it forces everything to go into a cell.
Exactly. To get past the endodermis and into the central vascular cylinder, the stele where the xylem is, solutes must cross a plasma membrane. Either they entered the simplest pathway earlier, near the root surface, or if they traveled via the apoplast, they're forced to cross the plasma membrane of an endodermal cell itself.
So the Casparian strip guarantees that every single ion or molecule entering the vascular system has passed through the selective filter of a living cell membrane.
Precisely. It ensures selectivity. The plant controls what gets into the xylem. It also prevents solutes that have been actively transported into the stele from simply leaking back out into the cortex via the apoplast. It allows the plant to concentrate nutrients in the xylem sap much higher than in the soil.
A very clever anatomical control point. Okay, so ions have crossed a membrane, entered the symplast, past the endodermis. How do they finally get into the xylem vessels themselves, which are dead cells?
That final step is called xylem loading. Ions move symplastically through living cells, like the pericycle and xylem parenchyma, right next to the xylem vessels. These living cells then actively transport the ions out of their cytoplasm across their own plasma membrane into the cell wall space surrounding the dead xylem trachery elements.
They pump them out into the xylem.
Yes, it's a controlled release or efflux. These xylem parenchyma cells have their own set of transporters, including proton pumps to energize the membrane, and specific channels and carriers that mediate the efflux of ions like potassium, chloride, nitrate, etc., into the xylem sap.
Are there specific examples?
Yeah, for instance, there's a potassium channel called SCOR that is specifically located in these stiller cells and is responsible for releasing K+ into the xylem. There are specific anion channels too. And importantly, this efflux process is regulated.
Regulated how?
For example, under drought conditions, the hormone ABA signals these cells to reduce the activity of channels like SCOR. This helps the plant conserve solutes and thus water in the root, rather than sending them up to the potentially wilting shoot. It's all tightly controlled.
Wow. So from the soil through the root, past the Casparian strip filter, and then a final regulated loading into the plumbing system. Quite a journey.
It really is.
Okay, so let's recap. We started with the basic forces, chemical and electrochemical potentials driving passive movement downhill, and the need for energy for active transport uphill.
Right. Then we saw how the plasma membrane H plus ATPase, the proton pump, is key in plants, setting up a strong negative membrane potential and a proton gradient, the PMF.
And that PMF then powers a lot of secondary active transport via symporters and antiporters, moving nutrients in and waste or toxic ions out or into the vacuole, which also has its own proton pumps.
We touched on the main types of transport proteins, the fast, gated channels for passive flow, the slower, specific carriers handling facilitated diffusion and secondary active transport, and the ATP-fueled pumps for primary active transport.
And the sheer diversity. Transporters for nitrogen compounds, cations like potassium and sodium crucial for salt tolerance, calcium for signaling, anions like phosphate, metals, even water channels moving things like boron and silicon.
And finally, we put it together in the root, seeing how the apoplast and symplast pathways work, how the Casparian strip acts as an essential checkpoint forcing membrane transport, and how xylem loading is a final, regulated step releasing ions into the transferation stream.
It really drives home how absolutely essential this constant controlled movement of solutes across membranes is for everything a plant does growing, getting food, dealing with stress. Understanding this, down to the specific proteins involved, really opens doors for things like improving crop nutrition and resilience.
It's truly fundamental. When you look at a plant, it's easy to forget the sheer complexity of the molecular machinery operating constantly within every cell, managing this flow of ions and molecules that ultimately allows the plant to build itself from simple components in the environment. Thinking about how this precise regulation, this dance of transporters, underpins all the plant life we see and depend on is quite something. What other hidden complexities are driving the visible world around us? Makes you wonder.